Review of Chapter 12, Cell Cycle and Mitosis. If you're wondering where's Chapter 11, we're skipping Chapter 11. Don't worry about Chapter 11. Now, Chapter 12 is on the cell cycle. The first thing you should be looking at, well, before you look at page 231, we talk about, in our notes, bacterial cell division, which is known as binary fission. With binary fission, it's rather simple. The bacteria replicates its DNA and then splits in half. That is shown on page 237, figure 12.11. Now, that's pretty much all I say about prokaryote cell division. The rest is eukaryotic cell division. Now, Roman numeral 2 of our notes, I go over the basics of eukaryotic chromosomes, page 230 of your textbook. The DNA in your nucleus is partitioned and packaged by proteins into a substance called chromatin. So chromatin is made of DNA and proteins. Chromatin is what chromosomes are made of. Now, what you see on page 230 is the chromosome before DNA replication, and then the chromosome after DNA replication. So, what we have, that's the centromere, and these are the chromatids. That's what the chromosome looks like before DNA replication. Although you don't see this under the microscope, this happens on the molecular level. Now, after DNA replicates, it looks something like this. The centromere is still the centromere, but these are now called the sister chromatids, and they are genetically identical. Now, of course, during cell division, they're going to separate so that each cell gets one of each chromosome. The only reason a cell would replicate its DNA is so that it can divide. Now, Roman numeral 3 is an overview of the cell cycle. You should be looking at the pie chart on figure 12.5 of your textbook. Now, most of the pie chart is surrounded by interphase. That's because that takes the longest time. Interphase is the entire time that the cell is not dividing. Okay, it looks like an ordinary cell. The nucleus is there, the chromosomes are not visible, you'll see a nucleolus, you won't see spindle fibers or anything like that. It just looks like a normal cell. So what's all going on during interphase is something you don't normally see under the microscope. Now you'll see the interphase divided into G1, S, and G2. The G stands for gap. Now the first gap is simply kind of a recovery after the previous cell division. Not much to tell you there. S, the S phase, is that DNA synthesis. It is during the S phase that the chromosomes produce the sister chromatids. Okay? Once a cell is entered into S phase, it's destined to divide. So, the G2 phase of interphase is more preparations for cell division. And that's interphase. The mitotic phase is the phase of cell division. And you'll see in this pie chart that it's divided into mitosis and cytokinesis. Mitosis really is the division of the chromosomes in the nucleus. Cytokinesis is the division of the cytoplasm, the physical splitting of the cell into two cells. Now you'll notice on the pie chart that there is no solid line separating those two words. That's because one begins before the other is finished. They overlap in time. Now, we cover mitosis in more detail from pages 232 and 233 of your textbook. Mitosis is going to produce visible changes in the cell. You'll study this in lab. Now, even though your book mentions prophase, prometaphase, metaphase, anaphase, and telophase, I'm going to omit the phrase prometaphase. I'm just going to cover them as prophase, metaphase, anaphase, telophase. Now, during prophase of mitosis, the chromosomes become visible. The spindle fibers start to appear. The centrioles start to move apart. The nucleolus disappears, and the nuclear membrane will also disappear. Okay? Visible changes. Metaphase is when the chromosomes line up, okay, in the middle of the cell in what is called a metaphase plate. Again, refer to the pictures in your book. Anaphase is a separation. That is when the centromere will split, the sister chromatids will separate and be pulled to the opposite ends of the cell. Telophase is kind of like the reverse of prophase. Everything that happened in prophase unhappens in telophase. Basically, the two new nuclei are starting to form in the new daughter cells. But notice that cytokinesis is happening at the same time as telophase. That is the best way to recognize telophase when you see it under a microscope in lab, is that it is happening at the same time as cytokinesis. Speaking of cytokinesis, that's the next Roman numeral in the notes. Oh, sorry. No, it's in the same Roman numeral, sorry. Cytokinesis is the splitting of the cytoplasm into two daughter cells. It's different between an animal cell and a plant cell. In an animal cell, cytokinesis involves forming a cleavage furrow, a pinching of the cell into two. Figure 12.9 at the top shows you the cleavage furrow. Figure 12.9 at the bottom shows you the plant cell. Cytokinesis in a plant cell involves a cell plate, a growing of a cell wall separating the two daughter cells. Now that's an overview of the cell cycle. Now in Roman numeral 5 of our notes, we talk about control of cell division. And for this, we move all the way over to pages 242 and so forth in your textbook. Now, in your bodies, not all of your cells divide at the same rate. Some cells, such as the skin cells and the lining of the stomach, divide continuously. Some cells divide only when needed to repair injury, such as liver cells. And some cells don't divide at all once they're fully formed. That's referred to as amitotic. Nerve cells and muscle cells, once they're fully mature, no longer divide. Now, there are regulatory genes that control cell division in every cell. Now, the notes discuss certain things that are discovered by growing cells in culture. For example, normal cells require growth factors, chemicals to stimulate growth. Normal cells have what's called density-dependent inhibition, meaning that they will grow and fill up their space, and when they fill up their space, they stop dividing. They also have what's called anchorage dependence. That is, the normal cells have to be anchored or attached to a surface in order to divide. You separate them, they quit dividing. Now, these are all natural controls. Cancer cells, tumor cells, represent a loss of control. They are growing and dividing uncontrollably. They will keep growing when they fill up their space. They will keep growing whether they're anchored or not. They will keep growing whether they have growth factors or not. So, in your notes, I describe the differences between benign tumor and malignant tumor. Benign is non-cancerous, does not metastasize. Malignant tumor is cancerous and can metastasize. Metastasis is when pieces of a tumor can break off and go and start tumors elsewhere in the body. Chemotherapy and radiation for treatment of cancer is aimed at stopping cell division. Stopping cell division. And that's our review of this chapter.